Okay, in this video I want to further stress the importance of concentration differences and this idea of a semi-permeable membrane and creating a membrane potential and things like that. These are all very important. My instructor, when I took the course, believed that, if you could explain something about how these semi-permeable membranes and how these concentration differences led to, changes in potential, which allowed for the firing of things like neurons and allowed for signals to be moved back and forth, in the brain, that this was a fundamental concept that everybody should understand. So what I did was I put together some sort of exam style questions that, things people might be asked to explain on an exam. And they're just really fundamental concepts. But the first one says, why do concentration differences across a semi-permeable membrane lead to a membrane potential? So why do we get this membrane potential? So the way I would answer it is potassium ions are more concentrated inside the cell than outside the cell. Okay, remember I said previously that originally they believed potassium ions were the only ions that were moving, that were creating this or moving in and out of the cell, creating this membrane potential, but that turned out not to be true. And I'll talk about why that is in a few minutes. So potassium ions are more concentrated inside the cell than outside the cell. As they diffuse out of the cell, they leave behind negatively charged organic ions. So the inside of the cell becomes more negative. So as the positively charged potassium ions leave, the inside of the cell becomes more negative. Since opposite charges attract, an electrical force develops that tends to pull potassium ions back into the cell. At some point, the diffusion of the potassium ions due to concentration differences, so just regular diffusion, is balanced with the electrical attraction tending to pull them back inside. This is when the cell reaches equilibrium potential. So that's how it reaches the equilibrium potential. And the important things are the two forces that balance are this sort of diffusion force, this idea of diffusion due to concentration differences, that balances with the electrical force because, as I said before, the inside of the cell is becoming more negative, so the positively charged potassium ions, you remember potassium has a +1 charge, is going to start to be pulled back inside. Okay, it's going to start being pulled back inside. There's going to be a force that's going to want to pull those positive charges back into the cell. And once those two forces balance, we get equilibrium, and that's basically what the Nernst potential is for these ions. So these, if you were calculating the Nernst potential for potassium or sodium or chloride, you would be doing that to find out when these two forces balance, when there's no net movement. So the second question I have here is, what experiments were conducted to show the neuronal membrane was permeable to more than one ion? Okay, so what experiments were done to prove this fact? Remember I said previously that originally most of the researchers believed that these neurons were only permeable to K+ or potassium ions. And that turned out not to be true, and that turned out not to be true because of this very specific experiment here. Now, of course, this is a very intuitive description of the process. It leaves out a great number of details, but it would be enough, I believe, for an introductory course in neurobiology or neuroscience to explain it this way, without going into all the excessive detail that the textbooks often use. The experiments involved the squid giant axon. So the reason they used the squid giant axon was because it's really big, and it really allowed the researchers a lot. I mean, if you know anything, I mean, if you have an intuitive idea of what an axon or how big an axon is, they're quite small, and in this case, this provided something that was really easy to work with, plain and simple. So the experiments used the squid giant axon, and the axon was placed in a bath, and this bath, contained the normal ionic concentrations of the various ions. So it contained the standard concentrations that you would find under normal physiological conditions. So it had potassium, sodium, chloride, etc. The only difference being one of the ions was replaced with a radioactive isotope. So what they did was they took one ion, let's say K+ for this example, and they used the radioactive isotope. And the reason they used the radioactive isotope was because they could detect the radiation once the experiment was complete and determine, did K plus ions or potassium ions move into the axon, or they move out of it, or in this case move into the axon, because the bath is what contained the solution. So that's the reason they used the radioactive isotopes. And the axon was then allowed to sit in the bath for a specified period of time. At the end of this time interval, the contents of the axon were removed and tested for radioactivity. So just what I said before. The experiment was also performed by extracting the contents of the axon and filling it with a specific concentration of ions, to be sure that the ions were both leaving the axon and entering the axon. We wanted to make sure, basically what the researchers wanted to do here was they wanted to make sure it wasn't a one-way street. So meaning the ions could, say, enter the axon, but they couldn't leave the axon, or vice versa. They wanted to be sure it worked both ways. Okay? And the experiment was then repeated for each ion. So what they did was each time they chose a different ion and used the radioactive isotope of it and repeated the experiment. And they also did it for varying lengths of time. They wanted to vary the amount of time that the axon was in the bath, or the amount of time that the contents were inside the axon. And what was found by these experiments was different from what was predicted. So remember I said they mostly predicted that K plus ions were potassium ions were the only ones moving. And that turned out not to be true. However, the experiment did determine that the axon was indeed most permeable to potassium ions. But it was also permeable to other ions with different, and that each of these ions had differing levels of permeability. And that's where you get these permeability constants from the Goldman-Hodgkins-Katz equation. Each ion has a specific permeability. It could be 1, it could be 0.5, it could be, 0.4, and and those are experimentally determined values for these for the permeability. So it's important to kind of see like how these experiments were conducted and what they did to help us understand what was really going on.